At transcription. And the thing to understand with both transcription and translation is that a lot of this has to do with just straight logistics of how the physical placement of all the different components is going to work out in making these proteins. And that's because if you got a cell, remember that your cell has got a nucleus inside of it. And inside of the nucleus is your DNA. Your DNA is inside of the nucleus. And the DNA only has one copy of every gene. The proteins that you want to make, they're going to be out here in the cytoplasm. And you want to probably make lots and lots of proteins. You don't want just one protein, you need a whole bunch of them. And so because you need a whole bunch of proteins, it's both safer and more efficient to have multiple copies of the genes that are on this DNA so that you can make lots of proteins at once. The equivalent is, let's imagine that, I want to show you something in class. It's a lot easier for me to hand out copies of the work that I want you guys to do, rather than everybody coming up and looking at one class set where we're all just going to stand around a desk. It's a bottleneck. So we want to have lots and lots of copies of all of this information going out into the cell so that way we can make lots of proteins at the same time. Well, the thing that's going to distribute all of those copies is RNA. And so we have both DNA and we have RNA. And those have different purposes. When we talk about their purposes, DNA is long term. DNA will last a very long time. We can isolate DNA from tissue samples that are sometimes thousands or even millions of years old. It's a really long-term sort of a molecule. So it's long-term, it's strong, it's expensive energetically, and inside of the cell we have only got one copy of the DNA because typically all your chromosomes have just got that one chromatid, so there's one copy of every gene. RNA, on the other hand, it is more temporary, and one way that is manifested, and the biggest way, is that RNA is only single-stranded. So DNA is expensive because it is double-stranded, but it's kind of messy, but DNA is double-stranded, RNA is only single-stranded. So that basically means that RNA is half the cost of DNA because we have half as much material. And so RNA is basically, you can think about it as like the post-it note of information. If DNA is something like, the Constitution of the United States, where we only have one copy of the actual original Constitution, that's the DNA, RNA would be like the Xerox copies of those information that you find in the DNA. The other, and this is a big difference, is that DNA represents all the genes inside of the organism, which we can also call the entire genome. RNA, on the other hand, is much smaller, and it only contains the information for one gene at a time. So when we do transcription, we have much fewer enzymes to worry about. And the reason for that is that if we were going to do DNA duplication, we need to duplicate the entire chromosome, the whole thing. And so that means that we have to unwind the entire DNA molecule, and we have a bunch of work to do in terms of just physically separating the entire thing. If we're doing transcription, rather than duplicating the entire piece of DNA or the entire chromosome, we just want to open up that chromosome enough to take a little peek, and then we're going to let it go back together again. We don't have to keep it open for the entire length of the chromosome. So transcription is going to start with our piece of DNA. And this represents, like, imagine we got a piece of DNA, okay? So this is not the entire piece of DNA. This would be just like a stretch of the original DNA. So since it's the original DNA, we're going to call this the template strand, okay? Well, this template strand is going to have a whole lot of genes on it, and we only care about the information for one gene at a time. So on this template strand, you're going to have a part that we're going to call the actual coding region. And if it's the coding region, it means that's the parts of the DNA that have all the instructions for how to build the actual protein. These are all the directions for how you're going to build that protein, which is on that strand of DNA. Upstream of the actual coding region is an area that we used to think was totally junk. In this area over here, it had lots of names. We would say that it's non-coding. I mean, that's the simplest way to describe it. It's non-coding. It doesn't have instructions for building a gene. But we now understand that it's not just junk. It's been called junk in the past. This is really important. This is the regulatory region. It is also sometimes called the promoter region. And what this does is it is the switch for the gene. This area of the DNA determines when this coding region is going to be copied into RNA and when it will not. It decides when to turn the gene on and when to turn it off. And it's going to do that because different proteins are going to directly bind to this piece of DNA at particular regions that are going to cause this piece to actually be turned into RNA. And we usually designate those with numbers that indicate how many base pairs they are from the beginning. So right here at the beginning of this gene, there is always going to be the same nucleotide sequence, and that is AUG. AUG always stands for the beginning of a gene, or you could also think of it as like the start. This is always going to stand for the start of a gene. So if we look upstream from where that AUG is, there are two regions that are always going to be present. One is at -10 and one is at -35. So at 10 base pairs from the start of the gene and at 35 base pairs from the start of the gene, there is always going to be the same sort of sequence. And that sequence is going to bind proteins directly to the piece of DNA. And there might be one of these proteins, there might be 5 of these proteins, there might be 500. It depends on the gene. But every gene is going to have a certain number of proteins that are going to bind to these upstream regions. And these proteins are called transcription factors. And the job of these transcription factors is to recruit the RNA polymerase, which is going to actually make the complement of this coding region. So the RNA polymerase is what is actually going to make RNA. And unlike what we saw with DNA polymerase, it does not need a primer. It does not need helicase and topoisomerase to open up this gene. RNA polymerase is going to make RNA from DNA. It can open the helix by itself, but it needs transcription factors to become activated. So it's not going to do anything unless these transcription factors turn it on. So we're going to have, let's imagine in this gene there's like 4 different transcription factors. They will recruit our RNA polymerase. So our RNA polymerase will come in and it'll bind over here. And it's bigger usually than the transcription factors. And when all of the different transcription factors have been recruited, it will then turn on and it will move along this DNA template. And as it's moving along, it's going to make our piece of messenger RNA behind it, which means that after transcriptions actually began, you're going to have this piece of DNA. In the area where transcription is actively happening, it's going to be opened up. You're going to have like a little bubble right here. At that bubble, you're going to have your RNA polymerase. And your RNA polymerase, just like the other enzymes, it's always going to read the DNA in a 3' to a 5' direction. So let's imagine it's reading the top part of this helix. That means on the bottom would be 5' and 3'. And then your transcription factors would be somewhere upstream from the start of that gene. And they would have been responsible for turning that RNA polymerase on in the first place. And as it zips along this DNA, it is making RNA. And the RNA is just going to be kind of like trailing off behind it until the RNA gets to the end of the gene. And at the end of the gene, so that means that the coding region, it would have already passed up the coding region. That would have been back here. And let's imagine this coding region extends to this point. When the RNA polymerase gets to the end of the coding region over here, that means that it is going to be where there is a stop codon. And when it reaches that stop codon, the RNA polymerase is just going to fall off of the DNA. And when that happens, your messenger RNA, which you just generated, will now become present on the inside of the nucleus where this whole thing is happening.